Hi, and welcome to my OCR A A-Level Biology revision session with me, Christine. So today's lesson I want to look at genetic diagrams and specifically dihybrid inheritance. So let's just remind ourselves about monohybrid inheritance. So Gregor Mendel looked at pea plants and he used observable characteristics to identify that there were differences between the phenotypes. The more we've learned about chromosomes, the more we are able to now understand how these inheritable factors are passed on to the next generation. So if we have two heterozygous individuals being crossed together, we should get a ratio of 3:1 if it follows Mendel's law of independent assortment. And if we look at monohybrid crosses, we should get a ratio of 1:1 if we're doing a test cross where we have a heterozygous crossing with a homozygous recessive. Well, let's just remind ourselves about this whole process and the fact that we know that individuals will go through meiotic division to produce their gametes. So meiosis is about the production of gametes, and so therefore when you have diploid parent cells inside the reproductive organs of an organism, what will happen is through meiosis 1, the first reduction division, homologous chromosomes will be separated during that anaphase 1. Through meiosis 2, the sister chromatids will then be separated during anaphase 2. And this will result in haploid daughter cells being produced. So in this case, in the egg cell, we have N, which is a haploid cell, and the sperm cell, N, which is a haploid cell. Now, through sexual reproduction, that combination of that genetic material from the two parental sources, what we're going to get is those two individual N haploid cells are going to fuse together. We end up with a zygote, a fertilized egg cell, which now has a diploid number of chromosomes. That will then go through mitotic division to produce all the cells inside the body as it grows, and then those cells will differentiate. So depending on which egg and which sperm cell come together will give us one way in which promoting that variation, which is why you will see variation within families. So independent assortment, the way in which the chromosomes line up in metaphase 1 will have a part to play in the way that they are distributed in the gametes. They distribute themselves independently of one another. So that's the first way in which sexual reproduction will promote variation. We need meiosis to occur to produce our haploid cells. Well, the way in which they line up during metaphase 1, which side do they coordinate themselves, that is going to have an effect. The next thing that's going to have an effect is the way in which they cross over. So that's to do with prophase 1. When they come together as bivalents during prophase 1, that is going to result in potential variation. And then the last one is about which egg, which sperm are actually going to be fertilized. So when we look at, for example, animals and we talk about the egg cells and the sperm cells and that fertilization process, we know that we're taking our haploid and we are resulting in a diploid cell. Well, plants are exactly the same. They have an ova, the female gamete, and they have the pollen, which is the male gamete. So when we look at the inheritable features, for example, the phenotype, we focus on the yellow and round seed of the plant. So when we talk about the pea plant and we look at this and the phenotype is yellow and round, I can see, because in my diagram they've told me the genotype. I can see that yellow is the dominant trait, and I can see that round is the dominant shape of the seed. And how can I see that? Well, I can see that because I've got a capital Y and I've got a capital R. Now, if I were to look at the potential gametes that can be produced, I would get 4 possible combinations. Now, it depends in the exam question. They might throw a curveball at you and give you a random example of the recessive traits, but I'm going to tell you that the recessive traits are green and they are wrinkled. So when we look at the pea plants, they can either be yellow and round, or green and wrinkled, or yellow and wrinkled, or green and round. So how do they come together? Well, if we take two F1 generation, two heterozygous individuals, and we cross them together, our F2 generation will result in these possible combinations. Now, the one thing I would always say is when you are doing a genetic diagram, always come down and across, down and across, down and across. Now, I always make sure I identify if there's at least one dominant allele that I represent that dominant allele first. So as you can see, each of my potential genotypes, if there is at least one capital, I start that first. So you can see that we now have the genotype in the box as well as the phenotype. So this is what's known as a Punnett square or a Punnett diagram. What I'm doing is I'm representing all the potential combinations that could be a result of random fertilization occurring. And what that would give me is that would give me a 9 yellow and round, 3 yellow and wrinkled, 3 green and round, and 1 green and wrinkled. So my phenotypic ratio is that I should get a 9 to 3 to 3 to 1 ratio with the offspring that I collect. Now that's important because that means that is following Mendel's law of independent assortment. And therefore, I can see my 9 to 3 to 3 to 1 ratio in the sample that I have taken. Now, it's important to note that just like with a monogenic cross, I also would potentially want to do a test cross. Now, to do a test cross, remember, that is where you're going to cross an individual that could be homozygous dominant or could be heterozygous, and you are going to cross it with a homozygous recessive. So it's important that you note that if you have a heterozygous with a homozygous recessive, that you should end up with a 1 to 1 to 1 to 1 ratio. Now, they love to throw you off in the exam slightly by giving you a random way of representing this test cross, where they could say to you that you have a yellow and wrinkled individual and you have a green and round individual. So if they were to cross a yellow and wrinkled seed plant and a green and round seed plant, you are still having heterozygous. So the first individual is heterozygous for the yellow color, but homozygous recessive for the wrinkled. The second individual is homozygous recessive for the color green and heterozygous for the shape round. So do note that if they are giving it to you in your dihybrid cross, that you identify which gene is heterozygous and which gene is homozygous, either dominant or recessive, depending on what they give you. We sometimes can make assumptions that they've given us a heterozygous and a heterozygous, and that they are heterozygous in one individual for both genes, but that is not always the case. We are still, if we have this yellow and wrinkled individual and a green and round individual, we are still doing a test cross. So I hope you've liked this video, and if you have, then please do click on the like button and subscribe to my channel. And if you haven't already done so, do check out my revision platform, www.aiqchat.com.